Hello, and welcome to Off Label with Altasciences, where we peel back the label in the drug development industry. The rise of microsampling as a technique has been a topic of interest in bioanalytical and clinical research communities for well over a decade, with advantages such as reduced sample volumes and improved patient comfort. Yet, despite these benefits and despite significant technological advancements, the widespread adoption of this patient-centric sampling in both clinical studies and general healthcare remains limited. But why? And how do we, the industry at large, change that? I'm joined today by our colleague and special co-host, Jeff Plomley, Scientific Director in Bioanalysis at Altasciences and passionate microsampler, as well as a very special panel of guests to tackle this topic. And I'd love for them to introduce themselves to you.
Hi, my name is Enaksha Wickremsinhe, and I work at the Gates Medical Research Institute.
I'm Katty Wan. I work for Pfizer in the clinical bioanalytics group under the translational clinical sciences.
Hi, I'm Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, and I'm really looking forward to this discussion today. Thank you for inviting me.
Jeff Plomley, Scientific Director in Bioanalysis at Altasciences in Laval, Canada.
Thank you all for being here today. Before we dive in, because I know we have a lot to cover about microsampling and patient-centric care, Jeff, maybe you can set the stage for our listeners. For those who may not be overly familiar with microsampling as a technique, what is microsampling and what are the general benefits, and how does it directly tie into innovative patient-centric sampling?
Well, the definition of microsampling really is often referenced as the collection of small volumes of biological matrices, typically less than 100 microliters. And it's used for the accurate determination of concentrations of therapeutic drugs, metabolites, biomarkers, etc. And it really represents a major component of the patient centric approach in healthcare diagnostics and drug development. In the vast majority of cases, it's dried blood that we end up with as a sample for analysis, but there are some recently introduced products from Capitainer in Sweden that also support dried urine and dried plasma.
Fantastic. When is microsampling a technique as an ideal choice? And are all assays compatible with the microsampling technique?
Well, if I could just backpedal one little bit, Lisa, there's just a few additional points I would like to make just to round out the initial question, if that's okay.
Sure, of course.
Okay, great. So, one point that I'd like to address is that although microsampling is not currently quite as mainstream as many of us in the audience here today would have predicted, the technique has gained significant momentum in the past decade for several reasons. And I'd just like to talk a little bit about those reasons. One is that advances in analytical instrumentation sensitivity have allowed us to hit our diagnostic endpoints with much less sample. And with that, typical volumes we use nowadays in the lab, specifically with mass spec detection, are between 10 and 20 microliters. And when we're talking specifically about implementation for regulated bioanalysis, microsampling device engineering needs to support accurate and precise blood volume collection that is independent of hematocrit levels. And so back in 2015, this was achieved by Neoterix with the launch of their Mitra device for fingerstick blood. And if you want to learn about the history and the evolution of that device, we have a podcast with Dr. James Rudge. You can find that in the Altasciences database. And there might be mention of a famous microsampler by the name of Neil Spooner as well in that interview,
I know him.
who played a significant role in bringing the Mitra device to market. So when the Mitra launched, that's really when we saw a resurgence in microsampling applicability, which really only increased when Tasso launched their on-body microsampler to further simplify collection. And that's achieved with the push of a button. And it also reduced discomfort that some users would experience with a finger stick, particularly beneficial for pediatric
Yeah,
populations,
I
right?
can imagine. Very
Yeah.
much so.
So this alignment of analytical sensitivity and device advances from Neoterix, Tasso, and Capitainer, just to name a few, microsampling now plays a key role in fulfilling a primary initiative of patient centric healthcare, which is giving patients the capacity to actively participate in their own treatment experiences. So we can ship these devices out to patients to collect microliters of blood in the comfort of their own home rather than the burden of having to travel to a clinic for milliliter volumes of blood by traditional venipuncture. And so, yeah, absolutely. And so this shift from in-clinic liquid blood samples to dried blood samples that can be taken essentially anytime, anywhere opens up an entire host of new opportunities for acquiring exposure data.
I would imagine it's much more stable too for shipping around the world and different climate
Absolutely, absolutely.
environments, things like that. So
Right. It gives us improved access to vulnerable and critically ill patients, for example, oncology and tuberculosis populations. It gives us opportunity for episodic sampling, for example, in anti migraine indications, which actually was the first volumetric microsampling application that we supported at Altasciences. But it's also important, I think, to note that we can enrich our trial by sampling for additional assessments, right? Such as measuring critical biomarkers, optimizing and individualizing drug dosage through therapeutic drug monitoring. We can more easily collect longitudinal data. And finally, the less invasive nature of microsampling itself has the potential to accelerate recruitment efforts, reduce dropout rates, and increase subject engagement. So, and as you mentioned, from a sample integrity point of view, there's the potential for improved stability for labile analytes, particularly prodrugs. And again, it does very much simplify the complexities that are associated with cold chain shipping and storage. So, I just wanted to round that out a little bit.
I love it. No, those are really, really good points. Obviously, it's an ideal choice. I was just curious, are all assays compatible to the microsampling technique, or is this something that you need to evaluate upfront?
No, I mean, workflows have been devised for the vast majority of compound classes. For example, MABs, antisense oligonucleotides, and of course, small molecules. There was a recent article published in 2025 by the Prati Group in, I believe the journal was Advances in Sample Preparation, and it nicely tabulates all of the applications as a function of device. And that includes the Mitra, the Tasso, the Capitainer, Hemapen, Hemaxis, etc. Now, the vast majority of detection is by LCMS, but there's also instances of immunoassay, qPCR, RT-PCR, ELISA, etc., even ICP-MS. In fact, I know Don Chase at Capitainer has been doing some amazing work on lead detection with the Capitainer B card.
This is very exciting. Well, since we're all gathered here to talk about innovative patient centric sampling, I'd love to hear about each of your interests and passions for it. So, why you feel so strongly about this topic, and I'd love to get your opinion from each of you. And Katie, I'd love to start with you.
My journey into microsampling began in the early days of dried blood spot sampling, largely motivated by the 3 Rs: replacement, reduction, and refinement in animal research. What truly fuels my passion for PCS or patient centric sampling is the impact it has on patients, especially children. During a patient appreciation day, I wrote a thank you letter to a caregiver whose child participated in one of our pediatric trials. Using a less invasive sampling approach that requires substantially smaller blood volume is a tangible way we can reduce stress and discomfort for sick children. That makes all the effort proudly worthwhile.
That's incredible. We're always very thankful for all the patients that participate in any of our clinical trials. And surely this must make it a lot more desirable and interesting for them. Neil, I'd love to get your view on this as well.
Yeah, absolutely. I think, we have a habit while we're busy providing healthcare or developing drugs that we're so busy moving forwards, we kind of forget who it's for, who the endpoint is, the people we're trying to improve the health of. And so, traditionally we demand they turn up in a clinical facility, often a long way from home, often at a time of day that's not very convenient to them, to collect a large volume of blood, most of which the analytical scientists throw in the trash.
Good point.
And this just doesn't feel right to me. We now have the technologies, whether for pharmaceutical clinical trials or for general healthcare or other times that we're taking biological samples from human beings, to take smaller samples, to take them in a more convenient time and place and process. And so, why aren't we doing that? And it's kind of let's give patients choice. It's about them. They're the people that are in receipt of the medicines, in receipt of the healthcare. It doesn't mean traditional phlebotomy goes away. That's part of the choice. Some people prefer that. Some people might prefer to go to their pharmacist. Some people might prefer to do it at home with a caregiver or themselves. Let's give people choice. And the exciting thing is that that then expands things. We can expand into more of the population in more locations. You don't have to be living near to a clinical study center. You don't need to be one of your typical cohorts that are recruited into clinical trials. We can diversify our clinical trials geographically and ethnically and in age wise and in many other ways. So the question is, why aren't we doing this? And that's kind of why the patient-centric sampling interest group exists, because change is always difficult. So why not bring together a bunch of passionate individuals and organizations who can work together to break down those barriers, to educate people and to listen to their concerns and then overcome them. So that's what gets me out of bed every day.
That's great. Well, it certainly seems like welcome change, but I love your point about giving patients options depending on, what's most appropriate for what we're looking to do.
So I think Katie, Neil, and I drink the same Kool-Aid.
Okay.
So, well, let me give you my take on this, right? So, routine and frequent monitoring is important for the timely identification and management of disease, right? But inconvenience, having to take time off from work to travel, finding childcare, to visit a clinic or a doctor's office, has been cited as the primary reason why patients were non-compliant or they withdrew from clinical trials, right? Because it's a burden. However, the COVID-19 pandemic fundamentally changed how healthcare is perceived and delivered, right? Including the acceptance of at-home diagnostic testing without the need for a doctor's appointment or to visit to a clinic. So this was significant progress for patient-centric healthcare, right? Including the collection of blood samples at a convenient location and time. Now, traditionally, we know that venipuncture performed by a trained healthcare professional using a vacutainer collecting 3 to 5 milliliters of blood has been the standard practice for over 75 years. But as technology and analytical methods have advanced, it is now possible to conduct most of these tests with only a few hundred microliters, maybe a few drops of blood, right? As you pointed out. Yeah.
Incredible. Yes, that's great.
So the new generation of patient-centric blood collection devices actually enable the collection of smaller volumes of blood conveniently without having to visit the physician's office or clinic. So I believe that these innovations can facilitate more convenient testing and monitoring of disease, resulting in better overall health outcomes.
So, Katie, I'm going to come back to you because, as Jeff established at the start, we have a method that reduces sample volumes, improves patient comfort. Why are we not using it more in general healthcare and similarly in clinical studies? What are some of the challenges and what solutions are already being used that continue to be developed to overcome these challenges?
Yes, I will use the Paxlovid program as example. And as you mentioned, yes, bioanalytical assays nowadays have enough sensitivity due to the new generation of instrumentations and also all the hard work done by our bioanalytical lab to improve sensitivity. So what we have encountered, the difficulties are not from bioanalytical assays. And we have used a Tesla M20 device across 11 Paxlovid studies and 3 second-generation Paxlovid studies. A key benefit of this approach was the ability to collect additional pharmacokinetic samples at home, supplementing venous PK samples collected on day 1 and day 5, thereby enriching the overall data set. So one of the primary challenge has been adapting the existing infrastructure largely designed for centralized trials with central lab workflows to support at-home sample collection. So that's the most challenge we feel. And we struggle quite a bit because that's really early on. That was in 2020, 2021. I think nowadays the workflow has become more mature and a lot easier to implement. Another challenge we faced also because of the early adoption is in global studies, the device regulatory approval constraints may result in exclusion of certain sites. But nowadays, all the device companies, they have been very diligent applying approvals from different regulatory areas. Lastly, PK bridging is compound specific, which remains an important consideration when implementing patient-centric microsampling strategies.
I'm sure logistics can be an interesting issue. So, Enaksha, why don't we talk about the training challenges?
Yeah, thank you. So, as we said, COVID laid the groundwork for patient centric healthcare on a global scale, right? However, this transfers the burden of performing the tasks now to the individual, right? Or the parent or the caretaker that will have to perform this task. This can be intimidating, right? So, therefore, training is key. And then also the ability and the level of comfort of each person can vary within a local population as well as it can vary quite drastically when you look at a global scale. Now, on the other hand, additionally, we have several different collection devices technologies, and each of these have different instructions. This could also end up being quite confusing. Manufacturers, I think, have done a pretty good job with depicting the procedures, usually using graphics, making it kind of easy to follow. However, these instructions still could be quite complicated with too many steps to follow. Looking at, if you're looking on a global scale, now you require translations and maybe different ways of communicating that information. A graphic may not be the best way to communicate that. So, you need to think of how training is done to communicate this message. Also, equally or probably equally important is instructions on how to return the samples, right, to the healthcare provider or the lab,
Yes. Right, Jeff?
which has to be easy and well described, right? So it can be done correctly. And also keep in mind the shipping efficiency will vary depending on the location, right? Whether it's metropolitan versus rural, or whether it's US or EU versus a lower or mid-income country, where resources may be scarce, right? Shipping efficiency is not the same globally. So kind of need to think of all of these when you're planning studies on a global scale. Establishing chain of custody is extremely important, as Jeff knows and all of us know, because that we need to ensure the integrity of the sample. Because if you can't ensure the integrity of the sample, your data is meaningless. Another interesting caveat that I want to add here is some people may not or do not want to perform self sampling or self collections and would prefer to actually visit a clinic or a doctor's office, right?
Good point.
This could be for many, many, many reasons, right? Some people may think that the official way of collecting it is in a clinic, and if I try to do that, I may, mess it up.
That would be me.
So, and some people may like that, adventuring out to a doctor's office, depending on, I mean, if it's a retired person living all by themselves, they would like to make that trip to the clinic once a week or twice a week to get that sample, whereas a mother taking care of 3 kids would opt to have her sample collected in a home setting, right? So we could think of the population, and there are multiple moving parts to
Certainly.
think of, right? So, I mean, training is challenging, as I said, and you need to think of all of these aspects when you're providing training to make sure that you get the right sample in a meaningful period of time.
Yeah, I mean, I think around the logistics do become complicated as well. Suddenly, rather than getting clinical trial supplies to your clinical centers and getting samples back from there, we're suddenly having to get samples to people's homes and relying on them to
Yes.
collect those samples and return those samples and turning those into multiple study centers. And in the pharmaceutical industry, that sample collection has to be anonymized. So how do we get that logistics to work while being anonymized? And obviously companies are solving these problems, but all these challenges add to the overall reluctance to change. We know what it's like. We all work in large organizations or have worked in large organizations where it's difficult to make a change, whether it's making changes for the analytical group, the clinical operations group, various groups involved within the pharmaceutical company or the contract research organizations, it's difficult to make change. And I think that's one of the big challenges. And so people always try and second guess and say, will the regulators like it? Will the data be accepted? And we know, there's been speakers from many regulators, including the FDA, who've stood up in public forum and said, we encourage this to happen, provided you validate your methods and you understand what's happening to the sample and you've thought it all through, we encourage you to use these approaches. So human beings are reluctant to change. I kind of live amongst people that are positive about patient-centric sampling, so it's quite difficult sometimes to see it from the other side of the fence.
Right,
And
of
that's what we
course.
try and spend a lot of time doing in the patient-centric sampling interest group, trying to listen to the people that have concerns and then figuring out, okay, how do we, rather than just telling them they're wrong, figuring out how do we get the data that helps just move it forward in different environments for different use cases for different barriers to moving forward. So yeah, it's human beings are conservative with a small c very often.
That's right. That's right. Well, you've convinced me for sure. So, Jeff, anything to add? Because you're often the recipient of these in the lab. So, anything to add on the logistics side before we move on?
Well, I just think that many of the device manufacturers are looking at engineering designs that mitigate sampling errors. It's all about time zero, right? It all starts at the minute that sample is taken and the quality of that sample. So you need to make sure it's volumetric, that it's not undersampled, that it's not oversampled. And anything the device manufacturers can do to mitigate those initial time zero sampling issues is a plus. And there are companies that are actively engaged in moving away from finger stick and more sort of
Yes.
on body, press of a button. Just take that entire process out of the hands of the patient. Anybody can press a button.
Exactly. Well, while I have you, I'm going to keep asking you the tough questions, Jeff. Talk a little bit about the approval status of the devices and the device dependency and how that's playing into this.
Honestly, I think Katie would probably be better to answer that because that's more of a regulatory thing.
Okay. Well, we'll get her opinion.
Yes, I will add a little bit. Of course, I want to say nowadays is much better than 5 years ago.
Okay.
And 5 years ago, a lot of devices are research use only.
I see.
So you all know my passion for pediatric use. A lot of devices are approved for adult use, but not for pediatric use. So you literally have to work with every single site and PI to present risk versus benefit to gain allowance for them to use it, even in the US. Some US sites are hesitating. Again, they have patients' well being in mind. So we understand that we really encourage all the device companies to use their network to do some trials or testings in pediatric population to gain that approval for the device. That will make the implementation a lot more easier in the future.
And if I may just say something, it can be complicated when you're doing a multi-site study because the regulations around the world are different and the approval around the world are different. And even, we've seen recently with Project Comfort, which maybe we'll talk about more later, even when something's got a CE marked and therefore is approved for use in Europe, when you go into the different countries, the way that is interpreted in the different countries is different. And so this becomes very tricky and quite a big overhead for companies to get involved in that. I think it'll get better as more companies do this, more people get familiar with devices, and more of the devices get approvals in different countries. But, we have statuses where we can't ship the blank samplers into certain countries within the European Union, even though the rules and regulations are the same in that country as they are in other European Union countries, where we can easily ship the samplers into that country. So hopefully we'll overcome some of those complexities in the future.
So if you've got a trial that's ongoing, do you encounter these challenges basically as the microsampling needs to be done across all these different geographical and sometimes different regulators? Is this an issue that you have to consider carefully up front, or how would you handle that? I open it to the floor here.
For our pediatric trials, we considered it upfront. Also, like some of the speakers mentioned, we use a hybrid approach. When it's possible, we do the patient-centric sampling. When it's not possible, we do an alternative sampling. For example, a lot of devices are not approved in Canada because they believe Canada have a smaller market value, just because less population. So what we have been doing, because Canada is an important country to provide clinical sites for us. So we literally have to work with each individual site, also rely on our regulatory affairs on the ground to work with local regulators. That will give you better possibility for success.
Yeah, I mean that's really the third challenge that we're talking about, isn't it? The device dependency, and it creates a situation where a study becomes highly dependent on a single product with very little flexibility if there are supply constraints. We have encountered that on one trial at Altasciences where one of the microsampling manufacturers encountered a hiccup, and weren't able to supply devices in a timely fashion. And that particular client decided to no longer proceed with microsampling, but rather go back to traditional venipuncture.
Okay.
So yeah, that is an issue. Um, One of the solutions that we provided was just to pivot to a different device, which, you can readily do because if you've developed a method on the capitator or the Mitra, those are readily transferable to other device technologies with the sorts of workflows that we use.
Okay. So
Um,
not a lot of lot to lot variability there, or you're
there
able to overcome that.
it's
Did I open a
better
can
it's
of worms?
better con
Sorry.
yeah. It's better controlled now than in the early days for sure. And I know Enaksha could speak specifically towards this, particularly with the baricitinib work that was done and published in bioanalysis.
I'd love to hear more in action.
Yeah, so I think just to add to the lot-to-lot variability, I think there were significant concerns on the lot-to-lot variability of the batch of devices. I'm not going to name names, but devices that were used. And one of the reasons is that most of these devices had a 18-month shelf life. So some of these trials take 3-4 years to run. So you are obviously going to get into different lots of material. And if you're looking at a 10 microliter sampling volume, a zero point 0.3 microliter difference could be pretty significant, right? If it's plus zero point 0.3 versus a minus zero point 0.3, it's about zero point 0.3 microliters, right? Whether it's 9.7 or 10.3, that it's going to be significantly different if it's not 10, right? So the variability is quite high off the bat. And then, that adds to your analytical variability. So we had encountered some issues with that with one of the devices. And I think, as Katie said, I think they're getting better now. But that's something we have to keep an eye out for.
But can you dial in specifications? So can you tell the manufacturers that a lot to lot variability must be within, plus minus X percent?
Yeah, at the time, Jeff, this was several years ago. At the time, I believe what we did was we purchased a huge lot of material and kept it reserved for our study. And so it was the same lot, and they would then issue a new certificate of analysis, when the first lot was up to its 18-month expiration, they would test the same lot and issue a new certificate of analysis, right? So technically, it was the same lot. It was stored under controlled conditions within their operations. And so I think that was the workaround that we ended up. May not be ideal because you got to invest a lot of money to buy an entire lot of material without
Right.
knowing how much you need.
Yes.
But that was how we decided to, go forward.
Katie, how does Pfizer handle that?
We in our validation, we evaluate lot to lot difference. Our very first trial, we waited about half a year to get 6 lots because they were not manufacturing that fast. But so far, from what we have seen, we haven't seen significant lot to lot difference. There was one time the manufacturer moved their location. They expand. So they moved to a new facility and produce new lots in the new facility. We even evaluate almost like a BE study, the manufacturer site change. And we do see some bigger bias than a previous lot to lot, but still well within 10%. So I think that gave us confidence that the manufacturer has good control with their product.
But even if it's 10%, Katie, that's going to add, like when you do your bioanalytical variability, you only allow 15%, right? That's what we found initially is I believe I received one lot that was 9.7 and the next lot was like 10.3, as I said before, right? So there's a 0.6 difference there. So that's 6% off the bat variability, right? One versus the other.
I guess the way I've always thought of it is, what is the variability we allow in our pipettes? And if they're a volumetric sampling device, they need to match what we consider we allow for our pipettes. That's the rule.
Yeah, that was also communicated. I don't think they were fully aware of that at the time. I think the pipette calibration is 3%, I believe,
Yeah,
right?
yeah. A lot of companies, it's around about 3%.
Yeah.
I think just for me, I think one of the other challenges to this being more broadly adopted in clinical trials is the fact that if we're going to do decentralized clinical trials and people can do more of the things involved in the clinical trials from home or somewhere that's convenient to them, the problem is we may be in a good place now for collecting the pharmacokinetic samples, pharmacodynamic samples with our specific biomarkers that we've developed. But many of the markers we're interested in are from standard clinical chemistry lists. And to begin with, not many of those analytes were available with assays that could be used with these home samplers. So what's the point of collecting your PK samples at home when you've got to toddle off to the clinic and collect your liver panel or your kidney panel at the clinic?
Right. You got to go there anyways.
Now, thankfully, people like Enaksha and others have been doing a lot of work, and gradually we're getting more and more of these devices, getting more and more of these clinical chemistry and hematology assays available with these devices, and that overcomes a significant barrier.
Great. So, Katie, let's go back to you. Very clear to me today that Pfizer deployed this very heavily for Paxlovid, but what is Pfizer's general viewpoint on the use of microsampling? How often is that being incorporated today? And are there certain criteria that sort of determine whether you're going to use it or not?
At Pfizer, we're committed to reduce clinical development cycle times by embracing innovations, including the incorporation of patient-centric element in 80% of clinical trials. So as an analytical group, we partner with study operations to develop a study-level playbook to support decision-making on whether patient-centric sampling should be considered. This decision framework is primarily driven by program needs and potential patient benefits, such as patient-centric sampling may be particularly advantageous in infectious disease studies, vulnerable populations, as mentioned by other speakers, pediatric, geriatric, and for compounds with long half-lives, where confinement can be reduced by enabling at-home sampling. The playbook also provides supporting resources for teams that elect to implement patient-centric sampling, including protocol and informed consent languages, central lab workflows for device distribution and sample returns, guidance on device malfunction, and adverse event reporting related to device use. And of course, information on the device approval status with local regulatory affair contacts to facilitate country-specific approvals. In parallel, we also partnered with a clinical pharmacology group to develop a dedicated bridging playbook This toolbook outlines a one-step and two-step bridging strategies applicable at different stage of clinical development, and includes a representative bridging study design with recommended sample sizes and acceptance criteria. Finally, we collaborated with the Pediatric Advisory Council, who will advise study managers assigned to the upcoming pediatric trial to engage our group early and consider patient-centric sampling as the default sampling approach once analytical feasibility has been established. In the last 5 years, we have implemented patient-centric sampling for PK across 11 clinical programs encompassing 24 clinical studies. So we are heading
You're committed.
pretty strongly.
Great. Okay, so let's pivot to more of the building of the momentum around microsampling. And Jeff, I know this is probably a bioanalytical scientist's dream come true here. This is innovative. This is everything you guys get up in the morning for as well. But obviously, there's a much broader implication here, right? For healthcare, for diagnostics, for larger shifts in patient experience and participation. So Katty, let's talk a little bit more from a clinical operations standpoint. And obviously, the patient centric benefits that you certainly observed, and that I'm sure you guys are looking to continue to observe.
Yes. We also recently just started, as Neil mentioned, a liver panel or other clinical standard panel. Can we do it with the at-home sampling? We have, I think, a clinical study set up. We already produced some data, very promising data. Good news is that a lot of device company also offer the liquid sampling platform.
Yes.
So those can be easily used for those standard panel. And also, I think what's most important is that the instrumentation for the standard, say, liver panel or standard, say, chemistry 15 panel is almost the same from our CRU, our research, clinical research unit. They handle a lot of phase 1 trials. They were able to use a smaller volume and run on a standard machine to get the results. So we're asking why the central lab cannot do that, because central lab has the same machine as ours. It's just that they are used to have a large volume. So their standard assay format is always a half a mil, but you really do not need that much. So this is, I think, a message we need to send to the central lab to encourage them accommodate a smaller volume so that we can do those standard panels using at home sampling device.
Okay, perfect segue. Enaksha, so she just stated it. What else would you document as the shifts in the industry that you believe are necessary to really have this become adopted mainstream?
Yeah, so changing from a Vacutainer to one of these novel devices causes a ripple effect through the entire workflow, right? From the start, from this time collect that sample to the time it hits the instrument to get the data. It's not the norm. It's very different, right? So it causes a lot of anxiety, causes a lot of changes to that workflow. And just to add to what Katie was saying with the clinical analyzers, the central labs are used to just popping their 3 milliliter tube on the conveyor and it runs, right?
That's right.
With microsampling, they need to use what they call a microtube or some. Yes, they need to
Yes,
transmit into a
microtubes.
smaller tube, and then then the workflow goes on. So there's a manual step there, which takes time and it's going to be expensive. Somebody has to do that, which is not the norm. So that's why I think they are a little bit hesitant to do that. But transformation must happen across the entire chain of events, right? Starting from the time of collection, shipping logistics, how do we manage all of that logistics? It's a different sample that's going to be shipped. Maybe it's shipped in dry ice. Maybe it's shipped, just without any refrigerant in there. Maybe it's just shipped in a shipping envelope. And then, the lab operations need to adjust accordingly. I think that's what's needed. Yeah.
Sure. So, Neil, what are some of the ways that the industry can actually promote the adoption of this? So, the patient centric sampling, who should really be championing this?
I think we all have to be champions, but because, as we talked about earlier, change is difficult, we founded the Patient-Centric Sampling Interest Group, which is a a not-for-profit group that brings together passionate individuals and organizations that believe in these technologies, but know that change is difficult. It was made even more difficult by the Theranos disaster some time ago, that made it even harder for people to believe that you could get high quality data from smaller volume samples collected in a way that puts the patient at the center. And so how do we work together to do that? So the patient-centric sampling interest group comes together to educate. We have a lot of webinars. We have a lot of working groups that are looking at what are the challenges, how do we overcome those challenges, how do we share publications, how do we publish our own data and move things forward. And hopefully by all of it, rather than a group of individual people and organizations facing the same way, why not all come together and face the same way and move forwards together? And we're very much led by what our members think we should be doing and how we should be doing it, and the voice of the patient about how to do it to move forward. And one of the really good examples of this is something called Project Comfort, which is a European Union funded IHI initiative, bringing together, I think, over 50 organizations across Europe, UK, and North America to look at the logistics of, as they call it, patient-centric microsampling for clinical trials and for healthcare. And so that is running in 7 different hospitals across, I think, 5 different countries, just looking at how we build on the logistics. We're not developing any new assays or any new devices, but we're demonstrating that the logistics can work and then sharing that information in an open access way. We're about a year and a half into a 3.5-year program, and the clinical aspects are just starting. But things like that are very exciting, and we run in-person and virtual events. We've got a series of virtual events coming up in April and May, where we're just working with the diagnostics community to ask what are the challenges and how do we overcome them. We're doing something similar in Africa. We're doing something similar in Australia. We're doing something similar with different professional organizations around the globe as to, what are the barriers? How do we overcome them? How do we move this forwards as a group of passionate individuals and companies? So it's very exciting. I think this is a key year.
Quick question, Neil. I'm very excited about the patient-centric sampling interest group journal. Can you tell us a little bit
Yeah,
about that? Yeah.
yeah, absolutely. So yes, people like yourself, Jeff, are always writing to me and saying, where's the best place to publish my data? And as it happens, within the patient-centric sampling interest group, one of our directors runs a publishing company, and one of the other directors runs a diagnostics lab. And so we've got together and we've launched the Journal of Patient Centricity. So it's going to be an open access journal, fully peer reviewed, and we have an editorial board. We've just received our first manuscripts for review. And it's not just for analytical work, it's for anything that is around patient-centric science to be published there. Very exciting. And the patient-centric sampling interest group are really excited to be part of that. And we look forward to that, just helping also to break down the barriers, as having a community newspaper that we can all go and read the latest information without having to search around hundreds of different journals.
Yes. Yeah. Looking forward to it.
Nice.
Thank you. Thank you for that, Jeff.
Okay, well, this is perfect because it wouldn't be off label if I didn't throw a curveball question out there to all of you. We've talked a lot today about the reluctance to change. So, what would be one thing each of you would say to those reluctant to adopt patient-centric sampling?
I just asked them to give it a try, to look at the different. We see communities, we see that can't access healthcare and can't access clinical trials, whether they have learning disabilities or whether they have rare diseases or they're elderly or they're children. Look at that, talk to those patients, talk to those carers and parents, and just listen to what they have to say and think about, put the patient first And yes, your job, we made it a little bit more difficult, or in some cases, a lot more difficult. But try and think of the bigger picture and what is happening for the rest of society. And the people we're hoping to treat with our medicines, what do they actually want?
Yeah. Okay. Enaksha, what would you say?
Yeah, so I think it's not a panacea, right? I think it's picking the right tool for the right task and then explaining it to people. I think there's a lot still to do with education and educating the broader population. That's probably one of the challenges. And as Neil said, Neil's group is doing a fantastic job in trying to reach all stakeholders in this and to provide information and share information and educate people. I think it's just providing the right technique, right opportunity for the right task, right? You can't throw everything at everything. You got to pick and choose what you want and then go with it. Yeah.
And Katie, I know you have an opinion.
I really want people to utilize the PCSIG that Neil is heading that organization, the resources in that website. It offers comprehensive references on how to do things, so it will get you started. My point is that right now, maybe we're the few early adopters, but based on all the benefits, we do believe this is going to be a mainstream in N years, maybe. But again, I will challenge our industry peers. Do you want to be, the late adopters when everyone else have this capability and you have to play the catch-up game?
Good point.
Or you want to go to that website, teach yourself how to get started, and maybe that will motivate you to get started because it's not that difficult. We have a lot of resource information on that website.
And Jeff?
Right. So I'm going to tackle this from the bioanalytical point of view, of course.
Of course.
And that is, in my experience, the bioanalytical workflow is actually simpler with dried blood microsamples than it
There you
is
go.
with liquid samples. And the reason for that is the sort of workflows that we use is essentially a solid liquid extraction where the majority of blood components remain on the substrate. So the extracts are actually cleaner, the workflows are faster, you have less issues with matrix effect. So it's a win win all around from the bioanalytical point of view.
So this has been super interesting to date. This has been fantastic. I love to look ahead though, and something that I love to do when I wrap up these conversations with my guests is ask for your crystal ball boldest prediction for the subject at hand. So I'm going to go around the table and I'm going to ask for what you think the future of microsampling holds. And Jess, I'm going to start with you.
Well, I mean, obviously, I'm going to tackle this from the bioanalytical point of view because I'm a lab nerd. So um, I'm extremely confident that, and I mentioned this before, that device advancements will continue with the aim of mitigating some of the data recording challenges, not only when sample is collected, but also when it's shipped. And I'm excited by this because Capitainer recently announced a next generation prototype in their portable card format that has integrated electronics that are powered by a bioenzymatic fuel cell. And this captures the time of sampling and records temperature and humidity exposure during shipping. It works with both their blood microsampling and their plasma separation cards. The data that's recorded is currently read out using near field communication to a smartphone, but that's just for demo purposes. But future iterations will allow for RFID readout directly into a limb system. And so, looking at the device, it promises to be rather foolproof because the sampling clock is passively activated when matrix, that is blood,
Yeah.
enters into the system. So there's absolutely no user intervention. So now temperature and humidity readings are recorded, and we know those are critical, right? Because that allows us to ascertain whether or not a sample was subjected to conditions where analyte stability might have been compromised. And that's something we examine during our method development at Altasciences. And we do that to establish guardrails for sample integrity. and therefore whether or not a particular sample can be analyzed. So now having the temperature and the humidity information embedded with the actual sample itself is going to be a game changer. Absolutely.
So I've heard a lot that this isn't necessarily just the bioanalytical scientists excited about this. This is what you're saying, Jeff. Obviously, it's a big part of it.
It solves a lot of logistical issues, right?
Yeah,
The
absolutely.
time the sample was taken, what happened during the journey of that sample. That is now all going to be embedded with the sample with the use of these bioenzymatic fuel cells.
Great.
It's
That's great.
incredible technology.
All right, Katie, you're up for your bold prediction.
Um, I predict the patient-centric sampling is gonna be probably replacing the traditional venous sampling maybe in the
Oh,
next decade.
okay.
I hope so. And then again, it brings a lot of benefit and infrastructure wise, where we can accommodate. And also, by the analytical part, we know we can do it. And now with the innovation of the devices, I think that's also add another layer of security in terms of sample chain of custody and the conditions. And also, I want to mention there are portable centrifuges we have used to generate liquid samples because, again, liquid samples are a lot easier to bridge. Some of the biomarker assays that require liquid samples. So, again, with all these advances of devices and facilitating tools, e diary, e journal, and all that, I think all these will come together and revolutionize the future PK sampling platform.
Very good. Enaksha, let's hear your bold prediction.
Yeah. So change takes time, right? Especially when we are trying to change a gold standard that has been around for over 75 years, right? Joseph Kleiner patented the vacuutenas in 1949. So it's more than 75 years. But I think we have made some very, very great strides in the right direction. So I believe that these technologies that enable convenient sampling will definitely have a positive impact globally and will eventually lead to better health outcomes for everyone. So I think my prediction is a healthier future. That I see people getting their testing or sampling done either self performed at home or maybe even at a local pharmacy or a grocery store while running their shopping errands, right? So I think, just one stop shop. I mean, that was one of the things that was also discussed. If people can't or don't want to perform this task at home, I don't remember the statistic, but in the US population, I believe 80% of the population lives 2 miles from a local pharmacy. So a large number of people have access to a pharmacy. They can, if we can have this set up at a pharmacy, they can go and get their testing done at a pharmacy. I may be a little bit off on that stat, but it was a significant number. So yeah, I mean, I think this is a reality. Yeah.
That's great. That's great. And Neil, some final words from you, please. Your bold prediction.
I think we're moving into a sweet spot for patient-centric sampling. I think there's the infrastructure and technological advances that Jeff talked about and Katie and Aksha. One of the other things that's happening that I've seen in some devices that had not yet made it to market is the ability to, within the device at the point of collection, do some of the analytical work. So some of the cleanup of the sample can be done at the point of collection, which is a game changer because then that sample arrives in the lab. A, it's more stable during transport, but B, it's kind of ready to go onto your instrument when it arrives. And so you have less need of what's going on in the wet lab. That's kind of exciting, and that's a bit of a game changer. Maybe 10 years off still, but I'm seeing devices being developed in universities along those lines. But also we're seeing healthcare systems talking about making healthcare more patient-centric in itself and bringing healthcare closer to the patient. The NHS in the UK has a big 10-year plan to do that, and patient-centric sampling is a key part of that in order for that to happen. So I think if these changes help in the healthcare system, it'll automatically overflow into the pharmaceutical clinical trial. And patients will start saying, well, why am I going to the clinic when I'm doing my regular healthcare from home? There's going to be that kind of question asked. So I think in that world, we're in a pivot point at the moment. And I think we've always had that graph of change with time. And people like ourselves have been the early adopters. And now it's gradually gaining momentum. And many of the situations that provide pressure or not on the outside are just starting to line up. So I think in 5 to 10 years, there will be an active choice of, do I go into a clinic? Do I collect it at home? Do I collect it at a pharmacist, as Enaksha was saying? And that's going to be wonderful for different patient groups in different parts of the country, different parts of the world, to have better healthcare and more inclusion into clinical trials. So it's all a good thing. So yeah, I think I'm hoping 5 years. I'm hoping to see it happen in my lifetime.
Well, I really appreciate getting to know all of you better today. Katty, Enaksha, Neil, and Jeff, you've dropped some incredible insights today. Thank you for sharing your expertise with us. It's been an absolute pleasure having you on Off Label with Altasciences. We're grateful for your time and for the valuable conversation. And until next time.
Thank you, Lisa. Thank you all.
Bye.
Thank you.
Thank you.
Thanks, everyone.